Hello everybody, as our class is cancelled for tomorrow, so we will have the class test on Thursday. So what I'm going to do, I have already posted the PowerPoint of chapter 5B on D2L along with the study guide. So I want you guys to go over the PowerPoint and read the chapter from the book, and I'm going to tell you a few important things which will help you in understanding meiosis. So because we have already covered mitosis, so it's easy to understand meiosis. So I'm going to talk to you about some of the important facts of meiosis, and you can go over things on the PowerPoint and from the book. And we will be discussing about meiosis maybe next week, because this Thursday we will have the test. So let me start the slideshow. Um, okay. So this is the PowerPoint. So basically, as we have already talked about, meiosis is the cell division by which the gametes are formed, and meiosis is a division by which gametes are formed. And you will see that there are some events in this process which are responsible for formation of unique kind of and unique and different gametes, which are responsible for our looking similar and dissimilar to our siblings as well as to our parents. So I will be talking to you about the most important things of meiosis. So basically, this is a division which starts with one diploid cell. So the important point to remember in meiosis is that before meiosis starts, there is an interphase. So in the interphase, everything is same like mitosis. So there's a duplication of DNA, duplication of everything. So interphase is same, but the difference is in meiosis we start with one diploid cell, and it results in four cells, and they are haploid, and those daughter cells, they are different from each other, unlike mitosis, where I have been telling you again and again, from one cell two cells will be formed, and those two cells will be genetically identical to each other as well as to the parent cell. And also meiosis involve two cell divisions. Okay, so the result of meiosis is four haploid cells, and they all are different, and these daughter cells, they're going to form gametes. Okay, this division basically has two phases, meiosis 1 and meiosis 2. Meiosis 2 is exactly as mitosis. If you understand mitosis, no problem in understanding meiosis 2. But there are some very important differences in meiosis 1. And meiosis 1 has four stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 again has four stages: prophase 2, metaphase 2, anaphase 2, and telophase 2. And they're exactly like mitosis. So I'm going to give emphasis on meiosis 1, because here are some differences. If you understand those differences, this process, this cell division will be very clear. Okay, so let's see what happens in prophase 1 and why it's different from prophase of mitosis, and what are those events which takes place in meiosis which is responsible for the formation of gametes which are unique. Okay, which are unique, which are different from each other, and that the formation of different kinds of gametes and the unique gametes is responsible for the genetic variation. So let's see what happens in prophase 1. Okay, in prophase 1, the most important thing, the other things will be same as the fragmentation of the nuclear membrane, disappearance of the nucleolus, spindle formation, chromosomes getting condensed, everything will be same. Now what is the difference? The difference will be in meiosis 1, in prophase 1 of meiosis 1, the homologous chromosomes will pair up. Now what are homologous chromosomes? Homologous chromosome, as you know, in humans we have 46 chromosomes, and these 46 chromosomes, they are in pairs. So in every pair there is one chromosome which is given to us by our father, another chromosome is given to us by our mother. So every chromosome, like for chromosome pair number 1, so the partners or the parental and the maternal chromosome number 1, they're going to pair up. Same for number 2, same for 3. So in the prophase 1 of meiosis 1, all the chromosomes, they pair up. And when they pair up, then the crossing over takes place. Now what is crossing over? Crossing over means the genetic exchange. Okay, so if you see this chromosome, so this is blue means from father, red means from mother. So you can see that this is a duplicated chromosome. This is also a duplicated chromosome, and these are the sister chromatids. Okay, so these blue are the two sister chromatids, these reds are the two sister chromatids. Now this one blue and this red, these are non-sister chromatids. Now when these chromosomes they pair up, there will be crossing over, which means that these non-sister chromatids, they are going to exchange the genetic material, which means some part of the red will become part of blue and some part of blue will become part of red. So you can see here how the genetic exchange takes place, and this process is called crossing over. So which means that some of the genes from mother's chromosomes, they're going to become part of the father's chromosome, and some genes from father's chromosome become part of the mother's chromosome, like this. So this is the way recombinant chromosomes, they're going to be formed. Okay, so this is the way. So this is a very important thing which takes place in the prophase 1 of meiosis 1, that's called crossing over, which is responsible for the exchange of the genes. And this process is responsible for the formation of unique kind of gametes. So this is the most important thing which takes place in prophase 1, which does not take place in the prophase of mitosis. Now what happens in the metaphase 1 of the meiosis 1? Now if you remember, we talked about in mitosis, all the 46 duplicated chromosomes came in the center. All the 46 duplicated chromosomes, they came in the center. But now, unlike mitosis, all 46 chromosomes are not going to come in the center, but rather 23 pairs are going to come in the center. Remember that the chromosomes are now in the form of pairs. And also remember, these chromosomes, they have already gone through crossing over. So they are recombined chromosomes. Okay, so these chromosomes which have recombined, now the chromosome pairs are going to come in the center. And you can see here, this is one pair and this is second pair. Suppose there's a cell which has two pairs. So you can see here, this is the chromosome pairs which are coming in the center, like this. Okay, so here you can see that now when these pairs, they come in the center, now there can be two kinds of arrangements. Like in this, you can see both the blues are on this side, and here both the reds are on this side, right? But in this case, you can see one blue is on this side, for the other pair blue is on this side. See, in this combinations, both the blues are on this side and both the reds are on this side. In this combination, blue of this is on this side, but the red of this is on this side. So how does one chromosome pair aligns is independent of the next one. Okay, and you will see that in the anaphase of meiosis 1, it is the separation of the chromosomes in the meiosis 1. Okay, in the anaphase 1 of meiosis 1, it is the separation of the chromosomes, rather the chromatids. So so you will see that it will result, because in the anaphase 1, it will be the separation of the chromosomes, and in the anaphase 2 of the meiosis 2, it will be the separation of the chromatids, because anaphase 2 will be exactly same to the anaphase of mitosis. So how the chromosomes align at this point is very important for the formation, how the gametes are going to be formed. So this arrangement, how these chromosomes are aligned, all the blues are on this side or all the reds on this side, how they are aligned, they are independent. So how does one pair aligns is independent of the second pair, and that results in the formation of unique kind of gametes. So this is the second event which is responsible for the formation of different kinds of gametes, and it is responsible for genetic variation. So as I just said, in anaphase 1 of the meiosis 1, instead of the separation of sister chromatids, it is the separation of the chromosomes. And in the telophase, so basically half the chromosomes will be on this side, half the chromosomes will be on this side. So if you started with a diploid cell which is 46 chromosomes, which means that you're going to end up with 23 chromosomes in the two cells, which means the two cells which are going to be formed, they will be haploid. So in meiosis 1, from one diploid cell, two cells will be formed which are going to be haploid. And after meiosis 1 will be meiosis 2, and meiosis 2 is going to be exactly same like mitosis. So remember that the result of meiosis 1 is two cells which are haploid, and now these two cells are going to undergo meiosis 2, which is like mitosis. So in the anaphase 2 of meiosis 2, it will be the separation of the sister chromatid. So which means that from two cells which are formed, which are haploid, each has 23 duplicated chromosomes. So each cell is going to form two cells, and each cell will have 23 chromosomes. So which means that the result of meiosis is you start with one diploid cell and you end up with 4 cells, and all are going to be haploid. So this is the main difference between mitosis and meiosis. So I want you to go over this PowerPoint, and you can see all these things. If you read it carefully, everything will be clear because everything is there in this PowerPoint. You can go over see the differences and how the gametes are formed. And when we will this Thursday we will have test, and next week we will go over this chapter shortly, and then we can proceed with our next chapter. So please, I want you to we really want to compensate for the loss of this lecture. So please watch this video carefully, go over the PowerPoint, read from book, don't waste your time, and I will see you guys in the class. Okay, bye.